Okay, so I'm going to quickly run through semi-conservative DNA replication. This is how DNA molecules make copies of themselves before a cell divides from one cell into two cells. And we need to know the stages, so I've split the stages into three parts. And if you've looked at the diagram, you will already have the color coding. This is the same coding for the nitrogen bases that we used when we're talking about the structure of DNA. So let's start here with this one. Here we have our DNA molecule. You can see our anti-parallel phosphate sugar backbones, and you can see the nitrogen bases. And in this case, we've got guanine, guanine, thymine, cytosine, adenine. That's the order, and then you've got the complementary base strands here. And again, I've drawn some little dotted lines in between the two to show that the nitrogen bases are being held together with hydrogen bonds. So this would be the DNA that's in the nucleus of what we call the parent cell. This is the original cell. This cell is going to divide into two identical cells, and before it can do that, it's going to need to make a copy of its DNA. And it's important that when the DNA is copied, it's exactly like this. The sequence of these nitrogen bases is exactly the same. You don't want DNA ending up with a change in this sequence of nitrogen bases, because that's a mutation. And mutations are almost always bad. So this is what happens first. This is an enzyme. I know it's an enzyme because it's ASE, right? ASE always tells me it's an enzyme. And in this case, it's helicase. Its job is to come in here and break the hydrogen bonds, so that when the hydrogen bonds are broken, these two strands of DNA separate. And you can see I did the phosphate sugar backbones here. I used a red map color because I need to keep track of where these are. So here is one side of the DNA molecule, and here's the other side. And in the meantime, right, these are going to act as templates, right? And a template is something that we use to make an exact copy. Okay, so this is going to be a template. I'm going to use this to copy, and we're going to use this to copy. And what you find is that in the cytoplasm of the cell, we have what we call free nucleotides. These are nucleotides. You know, you've got your phosphate, you've got your sugar, and you've got your nitrogen base. And some are adenine, and some are guanine, and some are cytosine, and some are thymine. And because of that rule of complementary base pairing, where A always goes with T and G always goes with C, this this nucleotide here is going to be attracted to its complementary base, just like this one here is attracted, the orange one is attracted to a yellow one. And so ultimately, all of these nitrogen bases on the original strands of DNA will be matched up with complementary free nucleotides. And what we end up with is this. You can see the original strand. Here it was acting as a template. Here's that original strand. And look at all these new nucleotides that have complementary base paired with the original strand. And then over here, we have the original strand and all the new nucleotides that have complementary base paired. And what that gives us is two new strands of DNA. And the hydrogen bonds have reformed. And this will go into one of the new cells, and this will go into the other new cells. And if you look at the sequence of nitrogen bases, green, orange, blue, yellow, green, orange, blue, yellow, green, orange, blue, yellow. So the nitrogen bases, the sequence of nitrogen bases, which is the genetic code, this ends up in one of the new cells, and this ends up in the other new cell. And so what we end up with is a complete set of identical DNA from the parent cell ends up into two of the new cells. Okay, that's a brief run through. What you need to do now, you'll see you've got these 3 boxes here. Okay, so I have a list of statements here. The only thing I'm going to tell you is 3 go in here, 3 go in here, 3 go in here. And you'll see these boxes. These 3 statements do not necessarily go with each other. These 3 do not go with each other. But when you cut these all out and stick them in the right section, this box will describe the first stage of DNA replication. This box will describe what's happening here with the 3 nucleotides. And this box will be the final stages. Okay? So this is what goes with the semi-conservative DNA replication. And just to remind you, it's semi-conservative. Semi means half. Conserve means to save. Half of each of the original strands of DNA have been saved in the new strands of DNA.